Hi, everyone. This is a screencast about phylogeny, which is the background you will need to do the lab for this week. So this is our second lecture for the week. So I'm not asking you to give any particular response. This time, usually I would ask you to take some notes, but I'm not doing that this time. So I hope you watch it because I think it'll help you with the lab and for understanding the material. So we're starting with this topic of phylogeny, which is how scientists organize and place on a tree the organisms that exist or have existed that demonstrates their evolutionary heritage. So, oops, sorry. There we go. So it's basically we need these principles. All life is related through a common ancestor. We already talked about that on Monday. So phylogeny is the evolutionary history of the relationships, So we sort of instinctively know some of this. Like a tree is more related to a bush than it is to a chimpanzee. We have this, sense of that. But how do we know how it all lays out? That's the study of phylogeny. So how this works is you create a phylogenetic tree, which is a diagram that kind of looks like a tree because it's branching out. So it's a dramatic reconstruction of diagrammatic, sorry, not dramatic. Could be dramatic. Diagrammatic reconstruction of the evolutionary history, and you lay it out in a way that looks like a tree. So a series of ancestor and descendant populations is called a lineage. So if you're looking from a species and looking back through time along the tree, that would be called the lineage. So it's a line drawn back through time on an axis. When a lineage divides in two, when you get that branching happening, that is called a node. So if you see the word node, that's where the branching is occurring. And that's when a new trait shows up in some way, whether it's a physical trait or even a change in the DNA. That would create a node. So as lineages split over time, you get this sort of branching tree that I can't really do it with my fingers because then they're all sort of branching from the same place, but you get the idea. Okay, so let's just look at this real carefully. You start off with a species, and as you remember, we trace back. So time going, in this case, to the left. This is called the lineage. And then here at the branch, that's the node. It's when a split occurs. Okay, and then along the new branch, you could list the new trait. And that's important for the lab because that's what they're doing is sometimes along the node, you have to put the new trait that emerged. And the lineages continue through the split. So if you wanted to look for the lineage of this organism, you would go, follow it back, and eventually get to the common ancestor. All right. So the common ancestor way back is called the root. So that's, makes sense. Tree, root, get it. The timing and the splitting of events is shown by the positions of the nodes on a time axis. So you always need to remember that we're talking about time as we move across the tree. So this is one that actually shows up in the lab. So if you pay attention, you get a little bonus for how this works. But so here we've got an evolutionary tree for humans, chimpanzees, gorillas, and orangutans. So these are all existing organisms. And what is their evolutionary relationship? You can trace it back. So humans and chimpanzees are very closely related. But they split off not from gorilla, let's be careful here, but a common ancestor shared by the gorilla and the human and the chimpanzee. So from this, the gorilla split off, and then this split off, and then that split off again to create human and chimpanzee. Okay, so if you started at this end, we have some common ancestor. From that, orangutans evolved, but then also a common ancestor of all three of these species branched off. Okay, and then we saw a new trait that separated gorillas from other things, and then a new trait that separated chimpanzees from humans. So I think this is common sense. I think it makes sense to all of you. But the question is, how do you pull it all together? I'm going to skip through that because I don't think we need this for the lab. Okay, so biologists determine traits that differ within a group of interest and then try to determine when these traits evolve. So you have to look at some kinds of traits. Now, originally, before we could look at DNA, we were only looking at phenotypes, right? We were only looking at things we could see as different traits, or even traits that may occur. Like a lot of times it's in embryo. A lot of times embryos keep some of the features that disappear as things get older, some features of common ancestors. But so things you could see, then we started being able to actually look at the DNA, and the lab goes into that as well. So you are not just looking at traits that you can see, but also as DNA patterns. Okay, so features that are shared by two or more species, that is considered homologous. So it's a new word. It's similar to that word we had when we were doing genetics with homogeneous, so don't get it confused with that. Homologous is a general word that means two things that are similar. So a homologous trait would be if you know both humans and chimpanzees have a large brain case, that is a homologous feature. So here's an example of vertebral common. So all vertebrates have a vertebrae column. That is homologous feature. But sometimes this can get a little confusing because sometimes there's this thing called convergent evolution where two traits emerge independently of each other. So, and it's not because they have a similar ancestor. So usually if you have two traits that are similar, it's because there's a similar ancestor. That's almost always the case, but every once in a while, this group over here develops the ability to glide, to have extra skin under its arm to glide. And then this other unrelated group over here evolves the same thing because it makes sense for their environment. It increases their fitness. And so we get these two things that developed independently, even though they look similar. And that is called convergent evolution. So, for example, bat wings and bird wings, they're not homologous. They did not evolve from the same common ancestor, but they are similar. Okay, so that's convergent evolution. So if you look at a bat wing and a bird wing, they actually have pretty different morphology and how the bones are structured. So again, they is convergent evolution where they evolved not from a common ancestor, but through adaptations and fitness to their environment. Okay, so this is what's really going to help with the lab. So here is a chart where we put a bunch of different organisms here on the left, and then say whether those organisms have these different features or not. And what you can do is you can actually see which ones are most closely related. Notice this one says outgroup. This is going to show up in your lab too. An outgroup is the one that is the furthest unrelated to all the others. So if you see something in the lab that says outgroup, you know that's going to be the first branch. Nothing else is going to branch out from, it's not going to keep branching. It's separate. So this outgroup is the lamprey, and it doesn't have any of these. But look, perch has one of these, but only one. So it is actually very similar to the lamprey. And this is what you want to look for. This is a key to doing this lab because the lab could get a little frustrating. You want to look for the organisms that have the most traits in common. So if we were to actually map this out, we would say, well, lamprey compared with a lamprey has 0 differences. Lamprey compared to the perch has 1 difference. So you can actually lay this out in a chart. So a crocodile compared to a lamprey has 5 differences, but a crocodile compared to a pigeon, where we go? Crocodile, pigeon, only has 1 difference. So if you look at the evolutionary chart that gets created, the crocodile and the pigeon are right next to each other. So again, this is a key for trying to do the lab. You could actually create a chart like that if you wanted to, if you're getting frustrated. But look for the things that have the most similar characteristics and start there and put them close together, and then work your way outward from there. You can start at the other end, but I found that much more challenging. It's easier to look for the similarities, to look for the things that have just one thing different. and put them together beside each other with just one branch in between, and work your way outward from there. Okay, that is where I wanted to stop for today on this. I will send a second screencast which is looking specifically at the lab and how you want to approach the lab. So stay tuned for that. Watch this because this is information you will need to know. All right. Have a good day.